Hello, my name is Lady Grant. I'm a biology student at the Community College of Denver, and my research involved finding the comparative abundance of bacterial taxa between free phosphate and aluminum phosphate conditions. Phosphorus is needed for almost every metabolic process in biological systems. This is why we add phosphates to our fertilizers to maximize plant growth. However, the overuse of these fertilizers is leading to an increase in agricultural runoff into our water systems. Now, the complicated issue is that most phosphorus pools are biologically unavailable. In tropical, highly weathered areas, phosphorus is largely found bound to metals such as aluminum and iron, resulting in an insoluble compound. Now, given that a large portion of these climates are currently being used for the world's agricultural needs, further investigation into insoluble forms of phosphorus need to be done to gain a better familiarity of how phosphorus mobilizes throughout tropical soils. The phosphorus cycle is the slowest moving nutrient cycle, with most phosphates entering the soil from the weathering of rocks. Now, as mentioned before, this research is focused on the role bacteria play in the phosphate solubility. So we designed this with two questions in mind. What kind of bacterial communities are present in these tropical soil types, and out of these, can they solubilize aluminum phosphate? Now, 8 soil samples were used in this experiment. The bacterial cells were harvested from the soil and placed into 3 different growing conditions. One with no phosphate, another with potassium and sodium phosphate, and a third with aluminum phosphate. After incubation, the DNA was harvested and sequenced using the 16S ribosomal RNA amplicon method. Now, as expected, more growth was observed on our free phosphate conditions. However, the species richness was observed to be higher on our aluminum phosphate plates. There are a few possible explanations for this occurrence. One is that competition was greater on our positive control, limiting the diversity of the plates, and 2 is that more bacteria than originally expected can live in low phosphate conditions. And one of the more interesting of the bacteria found was the genus Gradyrhizobium, because many of the species within that genus are known to fix nitrogen. The other 5 genus found have little to no ecological research done to our knowledge, with most available research pertaining to human health. However, this is a great starting point for further work. Our next steps would be to take the bacteria found in the aluminum phosphate conditions and administer more tests to see if they truly are solubilizing the aluminum phosphate, and to begin toxicity tests to see if these bacteria can also tolerate being in an environment with high levels of aluminum. And more research needs to be done to see what ecological role the other bacteria found play, along with a broader sampling of tropical soils to gain a solid foundation of the true dispersal of these bacteria. So out of the 2 questions this research was designed around, I'm happy to report that we have the answer to the first one. We know who's present in the soil types that were tested, and we can use this information to proceed to our next steps. However, this is just a small piece in a large-scale project, and I am excited to see what new data other microbiologists and soil scientists can bring to the project. Now, before I leave you, I'd like to thank the REX program for the opportunity to conduct this research. A big thank you to my mentors Tess Brewer and Dr. Noah Fear of the University of Colorado Boulder, and the National Science Foundation for funding our research. Thank you.